These single-celled animals, seen here under the microscope, have a large surface area in relation to their volume and have no problem absorbing substances such as food and oxygen over their cell membranes. Likewise, they can remove waste products like carbon dioxide over the cell membrane. Cells in many larger animals may be a long way from the surface of the body, and so there is need for a system which carries substances to and from the various cells in the body. Therefore, all these animals have a blood system. Blood is a liquid and is carried around the body in pipes, which are known as arteries and veins. Arteries have thicker walls than veins. The arteries and veins form a plumbing system in the body, rather like water pipes in a house. In order for the liquid blood to perform its function, it needs to circulate around the system. In the human body, blood is pumped by the heart, which can be seen in operation on this scan. Arteries carry blood away from the heart to all regions of the body, whilst veins return blood from the tissues to the heart. Mammalian blood, unlike some insect blood, is red in colour due to a pigment called hemoglobin. The different components of blood can be separated out simply by leaving a tube of blood, which has been treated to prevent it from clotting, in a rack overnight. After this period of time, the blood has separated into two layers. The top layer is a clear fluid called plasma. The bottom layer is red and opaque and consists of cells and cell fragments. Blood plasma contains a number of substances with different functions. Blood is liquid because of the water in the plasma. Minerals, such as salt, are carried in the plasma dissolved in solution. Dissolved salts in the plasma are important for maintaining a constant osmotic state in the body. The blood plasma is responsible for transporting many of the products of digestion of food. These substances include sugars, mainly glucose, from carbohydrates such as starch and complex sugars. Amino acids are the building blocks of proteins and are the final product of the digestion of proteins in food. Fatty acids and lipids are the products of digestion of fat in the food. All these substances are transported in the blood plasma to regions of the body where they are required as building blocks for complex molecules and structures. Water makes up about 90% of the plasma, and since carbon dioxide and oxygen dissolve in water, these gases are transported in the plasma. Carbon dioxide is carried both in the red blood cells and the plasma, whilst oxygen is carried mainly in the blood cells. Urea is produced by the tissues of the body as a non-toxic method of disposing of waste nitrogen. Antibodies carried in the blood are part of a complex system which recognises and destroys foreign particles in the body. The function of antibodies is linked to the action of white blood cells, as we will also see in a moment. The red blood cells which have settled to the bottom of this tube are responsible for the red colour of blood. These cells, seen here under the microscope, are essentially packages of haemoglobin. This red pigment exists in the blood cells in two forms. Blood which has just left the lungs is bright red in colour. Blood entering the lungs is dark red. This is because oxygen, which dissolves into the blood in the lungs, combines with the pigment in the blood cells to form oxyhemoglobin, which is bright red in colour. Oxygen is plentiful in the lungs, so dark red haemoglobin is converted to bright red oxyhemoglobin as it combines with oxygen. When the blood reaches the tissues where oxygen is low in concentration, it gives up its oxygen. So the bright red oxyhemoglobin is converted back to dark red hemoglobin. Red blood cells outnumber other cells in the body by about 600 to 1. The cells have no nucleus and are really just a membrane containing hemoglobin-rich cytoplasm. The characteristic shape of normal red blood cells, shown here by a plasticine model, is termed biconcave disc. This shape offers a large surface area for the exchange of oxygen. Also, no part of the cytoplasm is very far from the membrane. Therefore, the cell can operate as an efficient transporter of dissolved gas around the body. As the blood bathes the tissues, the red blood cells have to pass through narrow capillaries. These are capillaries from the tail of a tadpole. The smooth, flexible shape of the blood cells allows them to pass through these narrow spaces without blocking them. White blood cells are not actually white, but are colourless. The most common type of white blood cell is seen here. White blood cells have a protective function in the body. This cell is able to change its shape by extruding its cytoplasm rather like an amoeba. This enables it to engulf bacteria and other small particles. This is another type of white blood cell. It has a very large nucleus and it is capable of dividing very quickly. It is able to recognize foreign particles such as viruses and bacteria. Once the white blood cell recognizes a foreign particle, it divides and releases vast amounts of chemicals known as antibodies into the plasma. The antibodies released from any parent cell are directed against one particular type of foreign particle. They latch onto the particle, in this case a bacterium. The region of the particle to which they attach is called the antigen. The white cell recognizes the particle as foreign by its type of antigen. Attachment of antibodies usually results in the destruction of the foreign particle. Red blood cells are also coated with antigens, but the white cells are able to recognize them as self, and so do not produce antibodies directed against them. There are many types of antibodies in the blood and antigens on the blood cells. If an antigen called antigen A is present on the blood cells and there are anti-A antibodies in the plasma, the two will react and cause clumping of the blood cells. This could happen if the wrong type of blood is given in a transfusion. Therefore, blood is grouped in a system called the ABO system. A person in blood group A has A antigens on the blood cells and anti-B antibodies in the plasma. These do not react to cause clumping. A person in blood group B has B antigens on the cells and anti-A antibodies in the plasma. Again, there is no clumping. As a person in blood group O has neither A nor B antigens on the cells, the plasma can have both antibodies without danger of clumping. A person in blood group AB has both antigens and so has neither anti-A nor anti-B antibodies in the blood. This information can be used to work out which blood groups can donate blood to which other groups. In order to test which blood group someone belongs to, a card with anti-A and anti-B antibodies can be used. The antibodies are moistened with water. A blood sample must be taken. The handling of blood is dangerous, particularly with the threat of AIDS, so this test should not be carried out in the classroom. Drops of blood are taken from the patient and mixed with the antibodies on the card. Blood is also mixed with water as a control. This blood sample has started to show clumping in the presence of anti-A antibodies. This means that antigen A is present on the blood cells. There is no clumping with anti-B antibodies, so there are no B antigens on the cells. If we look at the table, we can see that the patient belongs to blood group A. When there is damage to the body and the blood is exposed to air, a soluble protein called fibrinogen forms fibres called fibrin. These fibres trap blood cells and cell fragments to form a clot. Bleeding from the wound eventually stops as the fibres build up a mesh which traps the blood cells and dries up to form a scab. When clotting occurs inside vessels or arteries, it causes a blockage known as a thrombosis. This is most serious when it occurs in a coronary artery, causing a lack of oxygen supply to the heart muscle and possibly a heart attack. If a thrombosis occurs in the brain, it may cause a stroke, resulting in the death of brain cells. Cholesterol in the blood, possibly due to an excessive intake of foods high in cholesterol, forms fatty deposits on the inside of blood vessels. This creates a rough base for fibres to collect and cause a thrombosis to occur. Blood has many other functions associated with both transport and protection. You may wish to discuss these further.